So thank you for kind introduction, Tommaso. So I'd like to thank Alain for giving me this opportunity to talk in this excellent conference. Although I'm not a neuroscientist, but I'm very much enjoying this meeting. So in my lab, we are focusing on the functional analysis of glycosaminoglycan chains in various tissues. So recently, we are interested in the function of chondroitin sulfate, representative glycosaminoglycan chain in the brain. So in this talk, I'd like to talk about role of glycosaminoglycan chain in the brain. So let me briefly introduce the structure of the chondroitin sulfate. Chondroitin sulfate is covalently bound to serine in the core protein and occurs as chondroitin sulfate proteoglycans. Chondroitin sulfate chain is a linear polysaccharide that is composed of a repeating disaccharide unit of glucuronic acid and N-acetylgalactosamine. During the biosynthesis, chondroitin 6-sulfotransferase 1, C6ST1, produces chondroitin 6-sulfation, whereas chondroitin 4-sulfation is formed by chondroitin 4-sulfotransferase 1, C4ST1. So recent many studies have proposed that the functional information of chondroitin sulfate chains might be encoded by specific sulfation patterns, so called sulfation code. So today, I'd like to talk about these three topics. The first one is how chondroitin sulfate sulfation patterns regulate the critical period plasticity in the adult visual cortex. And the second one is the potential role of chondroitin sulfate in neuronal migration and polarization in the embryonic cerebral cortex. The last one is the role of chondroitin sulfate sulfation pattern in neurogenic activities. So let's start the first topic. So as already mentioned, and also you know very well, the neuroplasticity, the ability of the brain to reorganize neuronal circuit based on new experiences. The plasticity is most evident during the limited time window in early life, so-called critical period, after which neurons become less plastic. So Kitagawa already mentioned about that balance between the excitatory and inhibitory circuit controls both onset and termination of the critical period. Especially the parabalbin expressing inhibitory neuron, PB cells play an important role in this process. So during the critical period, the chondroitin sulfate proteoglycans are diffusely distributed in the brain. After that, chondroitin sulfate proteoglycans condensed around the PB cells and forms a specialized extracellular matrix structure, so-called perineuronal net, in the adult brain. So Tommaso already mentioned about that. So removal of the chondroitin sulfate chains by chondroitinase reactivates the critical period plasticity. So based on this result, many researchers have believed that chondroitin sulfate chains are non-specific physical barriers that limit plasticity. However, in contrast to this idea, our hypothesis that chondroitin sulfate sulfation patterns may regulate the critical period plasticity. Its importance may have been overlooked in the previous studies because chondroitinase destroyed all chondroitin sulfate chains irrespective of sulfation patterns. To overcome this problem, we genetically manipulated chondroitin sulfate sulfation patterns in vivo by overexpressing chondroitin 6-sulfotransferase 1, C6ST1, because, as shown in the next slide, we previously reported that during the postnatal development of mouse brain, the proportion of 6-sulfation gradually decreased, whereas that of 4-sulfation progressively increased. As expected, transgenic mice overexpressing C6H1 showed an increased proportion of 6-sulfation and a decreased proportion of 4-sulfation, indicating that C6H1 transgenic mice retained immature-like chondroitin surface sulfation patterns in adulthood. So using this transgenic mice, we first examined the ocular dominant plasticity in the visual cortex, which is a classical example of the critical period plasticity. So we measure the visual evoked potential by placing an electrode on the right visual cortex and presenting a black and white stripe pattern alternately to the left and right eyes. So in mice, normal mice, the ratio of the left to right eye, VEP, is around 2, reflecting a contralateral eye dominance. If you patch or close the left eye for 3 days for critical period, the responses of neurons to the closed eye were greatly suppressed, which results in a decreased ratio of the left to right eye VEP. So we found that in C6H1 transgenic mice, plasticity normally occurs during the critical period. In contrast, after the critical period in wild-type mice, the eye closure did not induce a shift because plasticity is lost in the adult brain. However, the C6 transgenic mice show the shift following the eye closure, indicating that chondroitin sulfate C6H1 transgenic mice retain the juvenile-like plasticity in adulthood. So we wonder this persistent plasticity in transgenic mice might be caused by reduced perineuronal net formation. So as shown in this slide, most PB cells were surrounded by WFA-positive perineuronal nets in wild-type mice. In contrast, transgenic mice showed a significantly decreased WFA-positive perineuronal net as compared with wild-type mice. So 2008, Takao's group, in collaboration with Alain, reported that internalization of OTX2 by PB cells regulates critical period plasticity via its effect on PB cell maturation. The OTX2 produced in retina is transported to a visual cortex through thermocortical axon and internalized by PB cells. And however, how OTX2 selectively accumulated in PB cells. Because most PB cells were surrounded by perineuronal net, we thought that a sulfation pattern of perineuronal net might affect the incorporation of OTX2. So as shown in this slide, the OTX2 accumulated in PB cells in wild-type mice. In contrast, OTX staining was significantly reduced in transgenic mice, suggesting that accumulation of OTX2 is dependent on the chondroitin surface sulfation patterns of perineuronal nets. So when we look at in detail using a 3-dimensional reconstruction, in wild-type mice, WFA-positive perineuronal nets showed a meshwork structure surrounding PB cells. In contrast, some PB cells in transgenic mice form diffuse net-like structures that are stained by a CS56 antibody, which recognizes a 6-sulfation. In wild-type mice, B group T2 labeled thalamocortical synapses are embedded in wild-type in the meshwork of WFF-positive nets, whereas these synapses are not surrounded by a CS56 net, indicating that the diffuse perineuronal net rich in 6-sulfation cannot tightly surround synaptic contacts, which leads to diffusion of OTX2. So we finally examine electrophysiological properties of PB cells. So a basic fast spiking firing pattern were not significantly affected in transgenic mice. However, action potentials of in transgenic mice were significantly wider than that of wild-type mice, suggesting that because action potentials of PB cells become narrower during the postnatal development, we concluded that overexpression of C6ST1 prevents the maturation of the electrophysiological properties of PB cells. So let me summarize the first part of my talk here. In a wild-type mice, the WFF-positive perineuronal nets rich in 4-sulfation tightly surround synaptic contact, so that promotes the diffusion of, that promotes the incorporation of OTX2 into PB cells. This leads to maturation of PB cells and enhance the inhibitory circuits, which are required for the closure of the critical period. In contrast, the C6ST1 transgenic mice, some PB cells were surrounded by CS56 perineuronal nets rich in 6-sulfation. This diffuse light perineuronal net cannot tightly surround the synaptic contact, which leads to diffusion of OTX2. As a result, transgenic mice showed reduced inhibitory tone that is caused by impaired maturation of PB cells and retained the juvenile-like plasticity in adulthood. Therefore, chondroitin sulfate chains are not just physical barriers that limit plasticity, and specific sulfation patterns of chondroitin sulfate chains regulate the critical period plasticity through its effect on inhibitory circuits. So let's move on to the next topic regarding a role of chondroitin sulfate in neuronal migration and polarization in embryonic cerebral cortex. So let me briefly introduce the migration and polarization of cortical neuron in vivo. So after neurons are generated from neuronal progenitor cells, they become multipolar in intermediate zone. As they move to upper intermediate zone, their morphology changes from multipolar to bipolar, which extend leading process and trailing axons. The bipolar cells move to a cortical plate. However, what the physiological role of chondroitin sulfate is during the brain development before gliogenesis begins. So we first examine the distribution of chondroitin sulfate chains in the developing cerebral cortex at embryonic day 16. So as shown in this slide, the intermediate zone, the ventricular zone, so around 80% of chondroitin sulfate disaccharide is a 4-sulfation. In contrast, cortical plate contains a less 4-sulfation, and around 60% of chondroitin sulfate disaccharide is non-sulfation type. We also examine the mRNA expression of C4ST1, which is responsible for the formation of the phosphosulfation. So these results suggest that distribution of phosphosulfation well correlated with expression pattern of C4SD1. So therefore, we examine the spatial temporal expression of C4SD1 during the neuronal migration by transfecting these two plasmids, one expressed in M-cherry under the beta-actin promoter, and the other expresses GFP under the C4SD1 promoter. So C4SD1 promoter activity was already observed in a ventricular zone for 14 hours after transfection. 2 days after transfection, the most neurons moved to an intermediate zone. However, they still express GFP. In contrast, the 4 days after transfection, they no longer showed GFP, although the most neurons migrated to the cortical plates. So these results suggest that C4SD1 expression is transiently activated in the ventricular zone and intermediate zone, and is lost when neurons leave the cortical plate. So we examined whether the knockdown of C4SD1 may affect the neuronal migration. So 2 days after transfection with short hairpin RNA against the C4SD1, we collected a transfected region by laser microdissection, and C4SD1 expression was examined by a quantitative RT-PCR. So C4SD1 expression was significantly reduced in the area transfected. However, the initial phase of migration was not affected by C4SD1 knockdown. 4 days after transfection, control neuron migrated to a cortical plate. In contrast, migration was significantly inhibited by C4SD1 knockdown. The effect was successfully rescued by a overexpression of C4SD1. So 4 days after transfection, control neurons control neurons extended the axon toward an intermediate zone. When we stained a chondroitin sulfate chain by anti-chondroitin sulfate antibody, the axon extended along with a chondroitin sulfate-rich region. In contrast, chondroitin sulfate staining was significantly reduced in the area transfected with a short hairpin C4SD1. Control neurons in a cortical plate showed a bipolar morphology with a leading process and tailing axons. In contrast, C4SD1 knockdown neurons in an intermediate zone retained a multipolar morphology, suggesting that C4SD1 is required for multipolar to bipolar transition. So we finally tracked the morphological changes of primary neuron in vitro over 2 days using a time-lapse microscopy. So right one is a control neuron, and the left one is C4SD1 knockdown neurons. So during the 24 hours, both neurons can dynamically extend and retract the multiple process. We could not distinguish these neurons at this stage. However, after that, in control neurons, one neuron is selected to become axon and start to rapidly extend, whereas C4SD1 knockdown neuron cannot specify the axon, although it repeatedly extend and retract the multiple processes. So indicating that the C4ST1 is required for axon specification. So here is a represented image of neurons at day 3. So as mentioned, control neurons extend one long neurite, an axon, and we found that digestion of chondroitin sulfate chains by chondrogenase in control culture has similar effect with C4ST1 knockdown. So let me summarize the second part of my talk here. The C4ST1 and its product phosphorylation are highly expressed in intermediate zone and ventricular zone during the development of cerebral cortex. Phosphorylation produced by neurons itself creates a microenvironment that promotes a multipolar to bipolar transition during the migration. So let's move to the final topics regarding the role of chondroitin sulfation patterns in the neurogenic activities. So as already mentioned in the beginning of my talk, chondroitin sulfate is a linear polysaccharide that is composed of a disaccharide unit of a glucuronic acid and N-acetylgalactosamine. The building blocks can be substituted with sulfate groups at various positions, thereby producing a characteristic sulfation code. Major chondroitin sulfate contains a monosulfated disaccharide unit, the 4-sulfation, the 6-sulfation, the so-called A unit, C unit, respectively. And some chondroitin sulfate also contains a disulfated disaccharide unit, for example, the 4 and the 6 disulfated, so-called E unit. So we previously reported that chondroitin sulfate E, which is a predominant disulfate disaccharide E unit, possesses a strong neural outgrowth promoting activity toward hippocampal neurons. In contrast, CSA or CSC, reaching A or C unit respectively, showed a no or weak neural outgrowth promoting activity. So we thought that the functional receptor molecules receiving a sulfation code of chondroitin sulfate reside on the neuronal membrane surface. So at first, we attempted to uncover the mechanism underlying the chondroitin sulfate E-mediated neurogenesis using neuroblastoma cell line neural 2S cells instead of primary hippocampal neurons, because neural 2S cells differentiate and extend the neurite in response to serum deprivation. The possible contributions of neurogenic humoral factors being derived from non-neuronal cells are largely excluded from this neuronal differentiation system. So surprisingly, when neural 2S cells cultured on chondroitin sulfate E pre-coated substrate, neurogenesis was not observed. This initial observation lays a possibility that the insensitivity of neural 2S cells to chondroitin sulfate E due to defects of a particular cell surface molecules potentially sensing chondroitin sulfate E. So in the search for the candidate molecules by comparative gene expression analysis using a cDNA library derived from primary hippocampal neurons and neural 2S cells, we found that conductin-1, a GPI-anchored cell adhesion molecule of immunoglobulin superfamily, was not expressed in neural 2S cells. In addition, conductin-1 is also expressed in neuronal cell surface and interacted with multiple macromolecules, including a chondroitin sulfate proteoglycan, RPTP beta, phosphocam, and modulate neural outgrowth. So therefore, we thought that on hippocampal neurons, contactin 1 is expressed, and proteoglycan with chondroitin sulfate E structure binds to contactin 1, then leading to neural outgrowth. In contrast, contactin 1 deficient neural 2 cell cannot induce chondroitin sulfate mediated neurogenesis, indicating that contactin 1 is a neurogenic chondroitin sulfate E receptor, and that as a function is exerted through the chondroitin sulfate receptor mediated signaling pathway. So to examine the involvement of contactin 1 in chondroitin sulfate E mediated neurogenesis, neural 2 cells were stably transfected with an expression vector carrying a contactin 1 cDNA. The significant expression was observed in significant expression of contactin 1 and its cell surface distribution in established neural 2A contactin 1 cells were confirmed by quantitative RT-PCR and immunocytochemistry, respectively. So for analysis of chondroitin sulfate-mediated neurogenesis, the eight-way chamber slides were pre-coated with ornithine and then coated with chondroitin sulfate preparations, including chondroitin sulfate A, C, or E. Then the parental neural 2A cells or neural 2A contactin 1 cells were plated on the defined chondroitin sulfate substrate and incubated for 48 hours. The cultured neuronal cells were immunostained with anti-MAP2 plus anti-neurofilament antibodies. So as mentioned, in parental neural 2A cells, no neurogenesis was observed on chondroitin sulfate E substrate. However, as expected, in neural 2A contactin 1 cells, strong neurite sprouting was observed on chondroitin sulfate E substrate. So quantitative analysis measuring the length of the longest neurite of individual neural 2A contactin 1 cells cultured on various chondroitin sulfate substrata reveals that chondroitin sulfate E, as indicated by a green circle, showed a strong neural outgrowth promoting activity compared to polyornithine control, CSA, or CSC. Similarly, as judged by the increased fraction of neurite-bearing cells, strong neurite promoting activity was observed on chondroitin sulfate E, but not on chondroitin sulfate A or C. So notably, an application of functionally blocking anti-contactin 1 antibody effectively inhibited neurogenesis in neural 2A contactin 1 cells on chondroitin sulfate E substrate, so indicating that functional expression of contactin 1 is required for neurogenesis in neural 2A cells grown on chondroitin sulfate E substrate. So next, we examined whether the contactin 1 is also required for CSC-induced neural outgrowth of primary hippocampal neurons. Consistent with mRNA expression in primary hippocampal neurons, immunoreactivity for contactin 1 was observed in the majority of the MAP2 positive neurons in the culture. So as reported, the elongation of the prominent long neurite was observed in primary hippocampal neurons cultured on chondroitin sulfate E. As in the case of neural 2A contactin 1 cells, the treatment with contactin 1 functionally neutralizing antibody effectively inhibited CSC-mediated neurogenesis. However, the quantitative analysis measuring the length of the longest neuroid of an individual hippocampal neurons reveals that contactin 1 neutralizing antibody, as indicated by the brown circle, could not completely inhibit elongation of hippocampal neurons on chondroitin sulfate E as compared with a polyolefin control as indicated by blue circle. So these results suggest that there are additional regulatory mode of action for chondroitin sulfate E mediated neural outgrowth in cultured hippocampal neurons because chondroitin sulfate E binds to humoral factors such as brain-derived neurotrophic factor BDNF and midkine. So in fact, combined treatment with contactin 1 and BDNF neutralizing antibody, as indicated by a red circle, completely abolishes the promoting effect of chondroitin sulfate E neurogenesis, indicating that not only contactin 1 but also humoral factors such as BDNF is important for CSE-mediated neurogenesis in primary hippocampal neurons. So to further examine the receptor function of contactin 1 specific for chondroitin sulfate E, molecular interaction of contactin 1 with various chondroitin sulfate substrate were examined using SPR biosensor BioCore. Recombinant contactin 1 was immobilized on the sensor chip surface, and aqueous solutions of various chondroitin sulfate preparations were individually injected over the sensor chip surface to detect the direct binding. Unoverlaid sensorgrams disclose a clear binding of a chondroitin sulfate E. In contrast, chondroitin sulfate A over C showed less binding than did chondroitin sulfate E. So evaluation of the kinetic parameter revealed that chondroitin sulfate E interacted with contactin 1 with a significant affinity, whereas chondroitin sulfate A over C had 3 or 2 orders of magnitude of less affinity for contactin 1 compared with chondroitin sulfate E. So based on the kinetic data, the binding ability of chondroitin sulfate E to cell surface contactin 1 was examined by addition of fluorescent labeled chondroitin sulfate E to the culture media. So as shown in this slide, strong fluorescent signal was observed on the cell surface of neural 2A contactin 1 cells, whereas no detectable signal was observed on parental neural 2S cells, suggesting that direct interaction of chondroitin sulfate E with a cell surface contactin 1 is essential for subsequent actions leading to neural outgrowth on chondroitin sulfate E substrata. So several lines of evidence indicating that the contactin 1 mediated signaling is expressed as the activation of cytoplasmic non-receptor type tyrosine kinase fin, which regulate neural outgrowth. So to examine the involvement of fin kinase activity in CSE-mediated neurogenesis, we examine the fin kinase activity in neural 2A contactin 1 cells cultured in the presence of various chondroitin preparations or in the absence of chondroitin sulfate preparations. So as shown in this slide, the treatment with chondroitin sulfate E brought about a significant increase of fin kinase activity in a concentration-dependent manner as compared with non-treated cells. In contrast, treatment with CSA or CSC did not augment the fin kinase activity compared to the non-treated cells. In addition, no significant increment in fin kinase activity was observed in parental neural 2S cells, suggesting that CSE can act as an effective ligand for conducting one that stimulates the intracellular signaling pathway involved in neurogenesis. So in this talk, I show that chondroitin sulfate-mediated neurogenesis is exerted through conducting one mediated signaling pathway. So on the other hand, as I mentioned at the beginning of my talk, chondroitin sulfate is also reported to inhibit neurite outgrowth. Therefore, we suspect that the apparent contradictory actions of chondroitin sulfate in neurogenesis are attributable to its specific sulfation. And conductin 1 might be a bifunctional receptor interacting with chondroitin sulfate E and chondroitin sulfate X for promotion and inhibition, respectively, for neuroregulatory chondroitin sulfate. So in the search for the candidate chondroitin sulfate X molecule, by molecular interaction analysis using BioCore, we found that chondroitin sulfate D, which is characterized by the predominant disulfate disaccharide D unit that is structurally similar to but distinct from E unit, binds to conductin 1 stronger than chondroitin sulfate E. Because chondroitin sulfate D showed a weaker neurite outgrowth promoting activity than chondroitin sulfate E, so we hypothesized that chondroitin sulfate D might be an antagonistic ligand for conductin 1 and inhibit chondroitin sulfate E-mediated neurogenesis. So to examine whether the chondroitin sulfate D has inhibitory effects on the chondroitin sulfate E-mediated neurogenesis of hippocampal neurons, chondroitin sulfate E-mediated neurogenesis was compared in the presence of a very different amount of chondroitin sulfate D. So as shown in this slide, elongation of the prominent long neurite was gradually inhibited as concentration of chondroitin sulfate D was increased. So therefore, chondroitin sulfate D has inhibitory effects on the chondroitin sulfate E-mediated neurogenesis of hippocampal neurons. So to further involvement of conductin 1 in chondroitin sulfate D-mediated neuroinhibitory effect, so we examined whether the treatment of conductin 1 neutralizing antibody actually induced a neurite extension of hippocampal neurons of chondroitin sulfate E D substrate. So as expected, quantitative analysis measuring the length of the longest neurite of individual hippocampal neurons reveals that treatment with conductin 1 neutralizing antibody, as indicated by the open circle, indeed induces a neurite extension of hippocampal neurons on chondroitin sulfate D substrate, indicating that chondroitin sulfate D might be an antagonistic ligand for conductin 1, and conductin 1 might be a bifunctional receptor interacting with chondroitin sulfate D and E and D for promotion and inhibition, respectively, for neuroregulatory chondroitin sulfate. So let me summarize the last part of my talk here. So we show that involvement of sulfur fusion molecule conducting 1 is CSE-mediated neurogenesis. CSE, but not A or C, engage the conducting 1 and induce the intracellular signaling downstream of conducting 1, indicating that chondroitin sulfate E is a selective ligand for conducting 1. And chondroitin sulfate D has inhibitory effects on the CSE-mediated neurogenesis, suggesting that individual modification of chondroitin sulfate form a sulfation code that regulates the neurogenesis. So I'd like to thank these collaborators, especially in our lab, postdoctoral fellow in Shinji Miyata and graduate student Daiki Yasunaga and the staff member Tadahisa Misami. And also I'd like to thank other very helpful collaborators. Thank you very much for your kind introduction. Attention.